Hello and welcome to our symposium entitled The Human Connectome in Light of Evolution. Our aim today is to showcase some established and up-and-coming researchers in the field of evolutionary neuroimaging. Evolutionary or comparative neuroimaging is a relatively new field that should be of interest to human neuroimaging researchers who are looking for creative and leading-edge tools and approaches to understanding what makes human brains unique, both structurally and functionally. In recent years, the number of researchers involved in evolutionary informed analysis of human and primate brains has grown, as evidenced by the 2015 and 2018 Cortical Evolution Conferences in Las Palmas, Spain, the 2019 Brain and Behavioral Evolution Conference in Iriche, Spain, and the 2019 Comparative Neuroimaging Workshop in Dusseldorf, Germany, and the Prime DE Global Collaboration Workshop in London. The development of sophisticated techniques for imaging non-human brains, including postmortem brains, combined with the open data movement, is creating an opportunity for neuroimagers interested in human brain specializations like language, tool use, and social behavior, and to probe the anatomical substrates as well as the phylogeny of these abilities with novel comparative data sets. We hope this symposium helps you become familiar with evolutionarily informed approaches to connectomics, and provides you with perspective on the breadth of species which are being studied with comparative neuroimaging techniques and what new research questions these data sets will facilitate. My name is Katherine Bryant, and I'm a postdoctoral fellow at the Wellcome Center for Integrative Neuroimaging at the University of Oxford, where I use diffusion-weighted imaging to map the organization of white matter in humans and other species. Specifically, I examine the white matter organization of chimpanzees, macaques, and other primates in an effort to determine which features humans share with our primate relatives and which are novel human traits. For my talk, I'll be discussing this research and future directions for combining neuroimaging with an evolutionary biology framework and what kind of insights these data can provide. Michel Thiebaut de Schotten is the team head of the Functional Neuroimaging Group and research director of the CNRS at the University of Bordeaux. Previously, he co-authored with Marco Catani the Atlas of Human Brain Connections and helped co-found the Nat Brain Lab at King's College London and served as the head of the Brain Connectivity and Behavior Group at the Institute of Psychiatry in London and the Hôpital de la Salpêtrière in Paris. Michel will talk to you today about the revolution in neuroimaging made possible by open data initiatives, new comparative methods, and how evolutionary theory can be applied to comparative neuroimaging. Stephanie Forkel is a CNRS researcher in neuroscience at Bordeaux University and a visiting lecturer at King's College London. Her research is part of a Wellcome Trust-funded project studying brain connections and their impact on symptom severity and neurodegenerative disorders and cognitive recovery after brain damage from stroke and tumors. She's an associate editor for Cortex and a reviewer for over 20 journals and several funding agencies. Stephanie obtained pan-European training focused on clinical psychology and neuroimaging with an educational focus on postmortem and forensic sciences. Her research is capitalizing on the anatomy of stroke-induced neuropathologies like aphasia and anosognosia and the structural predictors of their recovery. Her work integrates the history of neuroanatomy with an emphasis on white matter connections. Stephanie will talk to us today about the human brain variability and how comparative primate neuroimaging can shed light onto shared and unique patterns of structural connectivity within and across species. Finally, Erin Hecht is a comparative neuroscientist and assistant professor at Harvard University, where her lab uses neuroimaging, histology, and behavior assays to study brain behavior evolution in humans, non-human primates, canines, and other species. Topics of study include social learning, tool use, domestication, learning and plasticity, and individual variation. During the quarantine, she keeps her lab skills sharp by making brain-shaped foods. Erin will be talking about evidence from comparative neuroscience and neuroarchaeological research on how human abilities for skill acquisition may have evolved.
Hello, I'm Erin Hecht. I would like to say thank you to the organizers for putting together this symposium and inviting me to participate. I'm happy to be here to talk to you today about plasticity and learning in ontogeny and phylogeny. So if you take a step back and look at our species from a biological standpoint, we're sort of weird. We're generally maladapted to the environments in which we live. We're not fast or strong. We don't have physical defenses or weaponry like claws or sharp teeth. And despite this, we've colonized the entire globe, including places where a hairless African ape really shouldn't live, like the Arctic. And we've accomplished this via individual problem solving, social learning, and culturally transmitted behavioral solutions to environmental problems. So it is through this anti-adaptation or maybe super adaptation that we have achieved our remarkable evolutionary success. So how did we get here? This image is a picture of stone tools used by chimpanzees in a Thai forest in the Côte d'Ivoire 4,300 years ago. And if you look at chimps today, they're using stone tools in basically exactly the same way. Meanwhile, if you take a look at our own species history in the last 4,300 years, we've gone roughly from the Bronze Age to sending fancy bits of technology into the furthest reaches of the solar system. So over the past few decades, there has been a lot of research about exactly what's different about how chimps and humans learn to perform actions that might account for this stark difference in our species-wide behavioral abilities. And it turns out that chimps and humans have notable differences in how they attend to observed actions. Chimps tend to attend mainly just to the result of observed actions, so what is accomplished through the action, whereas humans devote more attention to exactly how an action is carried out. So what objects and movements are used, how the movements fit together in space and time, and it is this more fine-grained attention to how actions occur that allows us to successively modify and improve these behaviors over time, leading to cumulative culture and achievements like exploring Pluto. So in my talk today, I will talk about some research about how these abilities might have evolved in the brain. First, I will talk about some studies using a phylogenetic approach, so comparing modern species in order to understand how brains have changed across evolutionary time. And the next, I will talk about some studies that take an ontogenetic perspective, looking at how networks change in an individual during one lifetime as learning occurs. So starting with the phylogenetic approach, this is something that I think we'll hear a lot about during the rest of this symposium, particularly from Dr. Katherine Bryant. So this is one way to study how brains have evolved. It involves comparing neural traits across related species in order to extrapolate their common evolutionary history. So in the case of our own species, our closest living relatives are chimpanzees. We diverged about 6 or 7 million years ago in the past. More distantly, we diverged from old world monkeys, including macaques. That occurred about 25 million years ago. So I'll talk about those 3 species today. And when considering complex behavioral abilities like social learning, culture, and tool use, an important thing to keep in mind is that complex behaviors generally rely on complex neural underpinnings. So the emergence of large-scale distributed functional networks like this is unlikely to be the result of a single random mutation. So this first image is a depiction of the mythical creation of Athena, who is supposed to have emerged fully formed from the head of her father, Zeus. This is probably not how brain evolution works. In order for new complex behaviors to emerge, they probably have to piggyback on on existing working circuitry that was previously supporting another function. So this is what is termed exaptation. For the behaviors we're talking about here, a good candidate for the exapted circuitry is frontoparietal circuits that integrate dorsal stream processing about vision for action and frontal circuitry for higher order action representations. We can trace the evolution of this frontoparietal circuitry all the way back to the earliest primates. So the first primates lived in the fine terminal branches of the tree canopy, where the price for misjudging the movement of your body through time might have been falling to your death. And to deal with these adaptive challenges, early primates evolved 2 new brain regions. One is area MT, which is involved in the visual perception of motion. And another is ventral premotor cortex, which integrates motor commands for the hands with visual representations from MT and related areas. In primates today, these 2 regions have these two regions have formed the basis of the dorsal stream, which has become enlarged and elaborated. So there are now many different regions that make up the dorsal stream. These regions are generally involved in understanding how or where actions take place. We can contrast this with the ventral stream, which involves a stream of regions that are involved in identifying what one is perceiving. So what body parts, which individual, which objects. And in macaques, an important point is that these two streams reach different regions of frontal cortex. The ventral stream terminates more anteriorly in ventral prefrontal cortex, whereas the dorsal stream terminates more posteriorly in ventral premotor cortex. We can study the connectivity of these circuits in chimps and humans using diffusion tensor imaging, which is non-invasive. It doesn't allow us to study connections at the cell-to-cell axonal level. So rather, we're looking at the anatomy of major white matter tracts. And if we look at some of the tracts that link dorsal stream regions with frontal cortex in chimps and humans, we can see extension of the third branch of the superior longitudinal fasciculus. So this is a tract that is shown in red in the chimp and human diagram and in green in the macaque diagram. So in chimps, SLF3 mainly terminates in ventral premotor cortex, but there is a visible projection into ventrolateral prefrontal cortex. And in humans, this balance is actually reversed. So SLF3 mostly terminates in ventrolateral prefrontal cortex with a relatively smaller projection into ventral premotor cortex. So this is notably different from macaques. So what might the functional significance of this difference be? One possibility is that it might allow for increased visuokinematic processing in ventrolateral prefrontal cortex. Another possibility is that it might allow for increased integration between the dorsal and ventral streams in prefrontal cortex. So these are studies that look at the anatomy of these tracts. What about their functional responses to stimuli? It turns out there are important species differences there too. For one, humans have unique sensitivity to 3D form from motion processing in the intraparietal sulcus. Humans also have a unique response to observed tool use in the anterior supramarginal gyrus. So these are both fMRI studies comparing humans and macaques. We did a study with chimps using FDG PET, which is a way to look at regional cerebral glucose metabolism. And this is one of my favorite studies that I've worked on because the result was just so noticeable, even without doing any sort of statistics. Humans show massively increased regional cerebral glucose metabolism in response to just observing a simple object grasping action compared to chimps. So there's increased activity both in dorsal stream regions leading into parietal cortex, but also in ventral stream regions. So I wonder if this might have something to do with the fact that chimps attend mainly to the end result of observed actions, which might be processed mainly in prefrontal cortex, whereas humans seem to spend more time processing the specific body parts, movements, and objects involved, which might be processed in these more posterior parietal and temporal regions. So these are comparative studies looking at differences across species, but what about differences within a species? So if this circuitry really is a functional adaptation, then we should see differences in these circuits in individuals within a species relating to differences in behavior in that species. So how can we study that? Now, bear with me, this might sound kind of weird at first, but one way to look at this in chimps is with mirror self-recognition. So most people have heard that chimps can recognize themselves in mirrors. They can, but actually not all of them can. Some chimps do this reliably, some never seem to recognize themselves in mirrors, and some show sort of ambiguous evidence of self-recognition. So I'm not trying to suggest that chimps have been selected to recognize their own reflections, but if you think about what recognizing your reflection entails, it has something to do with action representation. So if you are faced with your reflection, you see an animal in this weird shiny flat thing, And in order to not respond to it as if it's a conspecific, you have to recognize that the visual input that you're getting about the movements in this weird shiny flat thing correspond to the motor commands that you're sending to your own body and to the sensory feedback that those motor commands are causing. So in other words, you have to be able to compute correspondences between visually observed actions, motor commands that you're sending to your body, and sensory feedback that is resulting from those motor movements. So these are functions that might be important when you're monitoring your own or others' actions in the context of learning a new skill. And in this view, the ability to recognize oneself in a mirror is an epiphenomenon of neural adaptations for action understanding. So we looked at frontal parietal circuits in chimps that had been exposed to mirrors. And here's what we found. So this red tract here is the third branch of the superior longitudinal fasciculus. And we looked at this in chimps that showed no evidence of mirror self-recognition, questionable evidence of mirror self-recognition, and then clear positive evidence of mirror self-recognition. And you can see that there is a greater interior projection of SLF3 in the chimps that clearly recognize themselves in mirrors. So the pink areas in the diagram below are SLF3 terminations in ventral premotor cortex, and the blue blobs are terminations in prefrontal cortex. So one possibility is that this prefrontal extension of SLF3 is supporting increased integration between higher order motor representations, dorsal stream visual kinematic circuits, and ventral stream visual recognition circuits. So this might support the chimp recognizing itself in the mirror. So studying chimps is a great way to learn about our own evolution, but it can only get us so far because we diverged 6 or 7 million years ago, and a lot of interesting things happened after that split. So if we want to understand how human brains evolved after our divergence from chimps, we have to take another approach. So this brings me to the second half of the talk, where I'm going to talk about some studies using humans as model species for our recent ancestors. So this is work that has been done in collaboration with Dietrich Stout at Emory. Dietz is an archaeologist who studies how people learn to make and use Paleolithic stone tools. So this particular method of stone tool making is called Acheulean tool making. It involves repeatedly striking a core stone with a hammer stone to remove successive flakes. The action involves elements of recursion and hierarchical ordering, which has caused it to be proposed as a potential precursor to language. This is something that was used by multiple hominin species, not just Homo sapiens, for about 1 million years. So it was a major feature in human evolutionary history. In our first study in plasticity in this behavior, we trained 6 subjects very intensively over a period of almost 2 years, and they were scanned before, during, and after this training. So in this graph, the vertical lines are scans and the dotted lines are the average number of practice hours per week. So people were practicing fairly intensively. Along the bottom, you can see images of the types of tools that the participants in the study were producing. And then in the corner there in box C is an image of an actual Paleolithic stone tool. So you can see that people really did learn how to do this. So here's what we found. We found changes in FA in white matter and ventral premotor cortex, the anterior part of the inferior frontal gyrus and the supramarginal gyrus. We also found gray matter changes in the supramarginal gyrus. And when we did tractography on these regions that showed structural change, which is shown in this brain diagram here, we found that the network showing structural change closely overlapped with regions from previous functional studies. So in this diagram, sort of rainbow colored blobs are the network that showed structural change and tractography linking those regions, and the dots are foci of activation from previous functional studies. We also found that these changes in white matter FA were correlated with the amount of time that people spent practicing, and with a change in accuracy over time. So changes were coincident with increases in skill. More recently, we also did a second study involving a larger cohort. So this study had 20 participants. And we found that before training, FA in the interior part of SLF3 positively correlated with toolmaking skill. So before any training could happen, people who ended up being better, or people who had better initial attempts at the task had higher FA in this region, which I think continues to tie this region with an adaptive ability for toolmaking and social learning. Also, we found a reduction in interior SLF3 after training. So this correlated with learning trajectory after training. So we're still analyzing the data from this study. We haven't written it up yet. So hopefully you'll hear more about that soon. We also recently did a similar study looking at how brains change when they acquire modern technological skills. So we scanned people before, during, and after they took an introductory programming course. This is work that's being done by Suhas, a postdoc in my lab. So stay tuned for those results. To summarize, in humans, frontal-parietal circuitry, particularly the third branch of the superior longitudinal fasciculus, shows plastic change during the acquisition of evolutionarily relevant socially learned skills. This same circuitry shows progressive adaptation from macaques to chimps to humans. So recently evolved brain circuits support recently evolved behavioral skills. So this suggests that recent adaptations in the human brain are there to allow us to learn these types of skills. Over the past decade, there have been a number of studies that are sort of coalescing on this idea. For example, in humans, late developing regions overlap with recently expanded regions relative to macaques. Human myelination is extended in development relative to chimps. In humans relative to chimps, there seems to be reduced genetic constraint on our brain organization. And together, these findings suggest that distributed association networks like frontal-parietal circuits may have become somewhat untethered, allowing for an increased impact of experience and social context in shaping brain organization. So before I wrap up, I want to back up and look at this from another perspective. The big questions we're wrestling with here are how innate predispositions to acquire certain types of skills evolve. Or in other words, what is the interplay between heritable circuitry and experience-dependent plasticity? There's a way to look at this question that's kind of sitting right under our noses in a perfect natural experiment, and this is domestic dogs. So different breeds of domestic dogs have been selectively bred for different specialized behaviors. For example, hunting or herding or guarding livestock, or hunting by sight or by smell. Many of these behaviors rely on specific environmental experience during developmental critical periods. And some of these behaviors require protracted learning or training over a period of months or even years. So I wonder whether this evolutionary scenario might be analogous to things like how the human brain has become adapted to learn how to use tools or language. So this is something that my lab is delving into now. In our first study, we looked at a set of 63 scans from purebred dogs and just asked where variation occurs. So what you're looking at here is the result of a source-based morphometry analysis. This is an ICA analysis on gray matter morphometry data. So we identified 6 networks of regions that significantly covaried across the data set. So these are regions where the size of one region significantly predicts the size of other regions in the network. So they're regions whose anatomy co-varies across individuals. I won't delve into this too much here, except to say that there were interesting links between these networks and the behaviors that different breeds are specialized for. So for example, dog breeds that have been selectively bred to hunt by scent, their strongest association was with a network that involves olfactory components. Similarly, breeds that have been bred to hunt by sight, their strongest association was with a network that involves a cluster of regions that are involved in moving the body, moving the eyes, and spatial navigation. The strongest association with bird flushers and retrievers was also with this same network. So you could think if your job is to watch something that's moving across the environment and then figure out how to get your body to that thing and bring it back, those regions might be important for that job. Breeds that have been developed for explicit companionship, so these are like toy dogs or lap dogs, their strongest association was with a network that includes regions that activate to the perception of human faces or voices in fMRI studies. So an interesting note is that all of these dogs, as far as we know, were just house pets. They weren't working dogs. So what we're seeing here is probably the result of past behavioral selection in previous generations. So heritable innate differences in brain anatomy, not the result of experience. So that's the next question that we're going to try to tackle. How do experience and plasticity interact with these heritable adaptations? This is work that will be done in part by Sophie Barton, who's a grad student in the lab. So we're looking at dogs that are actively performing their breed-specific skill versus litter mates that, for whatever reason, are not performing that skill. So if you happen to have one of these dogs, or in fact any dog, even if it's just a house pet, we'd love to learn about it. You can participate even remotely, even just through the internet in the comfort of your own home during quarantine. So you could visit our website to find out more about that. And with that, I will say thank you again to the organizers and my co-presenters, and also thank you to the collaborators, lab members, and students without whom this work could not have been possible. If you have questions or you'd like to know more, please feel free to visit our lab website or email me. Thank you very much.
Imagine brain evolution as the next frontier. If you could travel through time as if each second were a million years, our planet appeared yesterday, the first tree grown 6 hours ago. Dinosaurs disappeared an hour ago, and primate would have appeared less than a minute ago, with only some of them walking on two legs less than 10 seconds ago. The first drawing has been drawn some 500 milliseconds ago, and as you can see here on the screen, the first carving of a human face was 250 milliseconds ago. This is a Brasserie, Venice in Paris. So a relatively short span of our recorded history happened only in the last 5 milliseconds. So we know nothing about our origins. We know nothing about the human mind at the prehistoric age. To explore this, of course, we can try to study fossils and extract those bones and look at the imprint of the brain, the imprint that brain left on the skull, as you can see here for the brain of Australopithecus. And thanks to MRI, we can make a digital version of how the brain of those prehistoric humans must have looked like at that time. Of course, we don't have that many remains of Australopithecus skulls, but we have a lot more of the Neanderthal, where you can do the same job and use the skull to reconstruct this endocast of the brain. And then you compare it with the human brain, and we can see that the Neanderthal didn't have the same shape of brain compared to humans. And you can see at the bottom of the slide, for example, the area are in green are the areas that you have to expand from Neanderthal to match brain from humans from today. So if you compare the shape from Neanderthal with the shape of the brain of modern humans, you can see in the chart on the right that they are radically different. They do not overlap. Although some humans have a shape of the shape of their brain that kind of look like the shape of Neanderthal. And what's beautiful of the study, in the study of Gunz et al., is they've been able to match specific genes in humans that are coming from Neanderthal that actually influence the shape of the brain to make it look like the one of Neanderthal. So that's extraordinary things that we can do with genes. Now we can actually extract from genes their age when they appeared on the phylogenetical tree and start projecting it onto the human brain. And the next decade, we'll probably see real maps of the phylogenetical evolution of the brain, of the feature of the brain, thanks to this projection from genes directly to the brain features. Another approach to study brain evolution is to compare species. And as you can see here on the left of the slide, this is the first drawing from Darwin of the evolutionary tree. By looking at similarities between species, Darwin has been able to draw these trees that explain how a common ancestor have been splitting to create new species. And you can see in the middle of this slide a picture that I took at the Natural History Museum in Paris, when in the front you have the skeleton of a sea lion that looked exactly very closely like the one of a cheetah in the back, or a rhinoceros like directly in the back. And that's because they have a common ancestor. Phylogenetically, they come from a species that follow, like according to the environment, have been splitting up in different species. So we're doing that in terms of anatomy, and we're particularly interested in the brain. One thing that is fascinating is that people in genetics, they're way more advanced compared to us in terms of building this evolutionary tree, and looking at what they've been achieving give us some indication of where we should be in a decade or so. So on the right part, you can see the evolution of a given gene, where you can see that human and rhesus monkey have a common gene that appeared 28 million years ago and started splitting into different phenotypes that characterize macaque and humans. And then later on, 6 million years ago, it split again to separate chimpanzees from humans. And then later on, you see that you have new phenotypes that appear inside humans. So those are within species evolution. And this demonstrates that by looking at inter-individual variability or difference within a species, we can actually assess recent evolution within a species. And you'll see that later with the lecture of Stephanie Forkel. Here we are mostly interested about the brain, and if you want to do comparative anatomy, you can compare how brain looks like across different primates. On the left, you have the beautiful work from Katja Oeher, who's showing all like 36 different primate species brains, and the first thing that you can see is they differ in size. But you know, don't get too excited. Just the size doesn't matter much. You need to look at it in relationship to the body size that you can see on the top right, which is different across primates. And if you do a ratio between the body size and the brain size, or the body weight and the brain weight, you can see that all species on the planet are more or less aligned on this big red line that you can see at the bottom right of the screen, except for humans that seems to be slightly outlier. So that means that humans compared to other species on the planet have a brain that is a little disproportionate compared to their body. If you want to look more precisely why it is disproportionate, you can see that in the literature there's been a long debate on whether or not the human frontal or prefrontal cortex is bigger proportionally than in other species. After this long debate that was between the year 2000 and 2010, we've been able to see that the relative size between the frontal lobe volume divided by the total brain volume is actually bigger in human than other species. Now, we know that the frontal lobe, or we hypothesize that the frontal lobe is very important to integrate associated information from the rest of the brain. And it can only do so by being highly connected with the rest of the brain. So some of the researchers investigated the connection in the brain, in the human brain, and compared it with macaque monkey. And by doing so, well, in this example, you can see the arcuate fasciculus, which is a main connection dedicated to language. We can see that the equivalent of the arcuate fasciculus in macaque monkey in humans is very small, and you have a lot of difference that are characterized here by connections that are in blue, where you can see that really the arcuate fasciculus in human is disproportionate. Really bigger. This can explain why, compared to macaque monkey, humans have a way more elaborate communication. Other researchers have been investigating the connective property of the brain by using functional connectivity. Now, functional connectivity is looking at the synchrony of the both signal between different brain regions. And the idea is we're looking at the synchrony of the neuron activity in the brain. Now, the neuron activity change according to what you do, whether you rest or think about different things. So in order to control for that, they exposed humans and macaque monkey to the same video. And by doing so, they synchronize their activity, their brain activity. And because their brain activity is synchronized between species, they've been able to compare the networks. And they found some specific networks in the human cortex that do not exist in the macaque cortex. And when they look at the comparison between where those networks are and the expansion of area, if we were to match a monkey brain onto a human brain, the networks that are not corresponding to macaque monkey networks are actually the regions that expanded the most when we compare a monkey to a human. Now, very recently, people have been trying to assess similarity and difference between human and monkey by using principal features, principal characteristics of functional connectivity. And here at the bottom left, you can see a preprint that is showing the principal gradient of functional connectivity in humans and macaque monkey. And by using this information and the other gradients, we'll be able to compare and carefully match the signature of the gradient in humans with the signature of the gradient in macaque monkey and do some translations that are based on this information. All this ultimately to have a very good match between the species, and I believe moving to the next step, which would be to do some very well-designed graph theory comparisons to try to see whether with evolution, as the brain of humans become more integrative and less modular, using less modularities than other species. Finally, another approach to study brain evolution is to directly use prehistoric artifacts and present it in the MRI scan to humans of today to decode their meaning. And that's what Emmanuel Millet has been doing. He used these ancient artifacts that you can see at the top, where the meaning of those lines was like unknown. So we didn't know whether this was accidental carving due to the elaboration of tools, or if that was like a symbolic representation from the prehistoric time. And by using functional MRI, he presented this carving to humans of today and compares their functional activation to the presentation of objects, scene, or ancient scripts, or words. And what he's been able to show is that those carvings are absolutely not accidental. Those are like true symbolic representation of object or ancient scripts. In sum, what I've been trying to show to you is that there is a wealth of approaches to study brain evolution with magnetic resonance imaging, and we should do it. You can use endocast imaging and use like skulls from the prehistoric age to estimate how the brain must have looked like. This is a direct approach. You can use genetic neuroimaging to project the age of genes directly onto the surface of the brain. You can use variability neuroimaging to look at differences between people of the same species in order to assess recent evolution. You can use comparative neuroimaging to find and estimate how our common ancestors a million years ago must have looked like. Or you can do what we call prehistorical neuroimaging to directly measure functional activation related to the presentation of ancient prehistorical artifacts. And why should you do so? Well, I hope to build a phylogenetical tree of the evolution of the primate brain, as you can see an artistic example on the right side. But if you don't believe that this is a reason good enough, just think about how understanding brain evolution will help us to understand the different strategy evolution has been taking in species to recover from disease or avoid disease, or recover from brain injuries that we could copy and match with the human brain for the future of brain medicine. With this, thank you for your attention, and I'll be happy to take your questions.
In this presentation, I will talk about the variability of structural connections within and across species as part of our symposium on the human connectome in light of evolution. There is a large amount of variability across human brains, which was revealed initially on a small scale by postmortem studies, and more recently on a larger scale with neuroimaging. In this talk, I will give you a brief overview of the variability in connectional anatomy and what this variability means for behavior, cognition, and evolution. But first, a spoiler alert, the take-home message from my talk will be that we are all the same, but we are different. And I want to give you an example of what I mean by that. What you see here are the faces of the women who won a Nobel Prize for physiology or medicine. The picture in the middle is the average of all these faces. While there are some features that resemble the individual faces, none of them look like the typical female Nobel laureate. But the face in the middle does resemble a face. It has all the features like a nose, 2 eyes, and a mouth. This is similar to what we do in neuroimaging, where we often study the structure and the function of the brain in a standard space. While this is a good procedure to make us comparable, when it comes to predicting individual behavior, and in particular clinical recovery, we often see that our models don't fit. So how variable are we? And while we don't have an answer to this question yet, I want to show you some examples of the variability. Here we see the variability within the same person, also known as intra-individual variability between the left and the right hemisphere. You can appreciate that the Sylvian fissure follows a different trajectory in the right hemisphere compared to the left. In the middle, we see a view of the ventral surface of the brain, where you can see the protrusions anteriorly and posteriorly, as well as the differences in the width of the frontal and occipital lobes. These anatomical features are often helpful when trying to work out if the MRI images were flipped during the pre-processing. On the right side of the slide, we can see the left and the right visual cortex as defined microscopically. The hemispheres are from the same person, but there is a clear degree of variability between the left and the right. These are just some examples of the variability within each and every one of us, but how different are we to each other? Brain morphology is not consistent across individuals. Using the visual system as an example again, this figure now shows the variability between people, or inter-individual variability. In addition, half of the population have an additional gyrus above the cingulate gyrus known as the paracingulate in at least one hemisphere, but not necessarily both. we have seen that regions with clearly defined functions, such as the visual cortex that I just shown you, but also the motor, the primary motor cortex, can be highly variable. The example that I'm showing here is the hand knob region that we often use to identify the primary motor cortex on axial postmortem slices or MRI scans. Now, while this inverted omega sign is often present, it can vary greatly in the presentation and can, for example, look like an epsilon, or in some people not even be there at all. Taken together, this body of literature indicates that there's a large amount of structural variability in primary cortical areas and associative cortices. But the relevant question is if these differences have a behavioral impact. One example is this study that had quite a media hype and showed a relationship between structure and function. In the London cabbie study, the hippocampal volume in cab drivers increased as they memorized all the streets of London and how to navigate them best. Although experience-dependent structural changes have been demonstrated in grey matter, there's little evidence for such changes in white matter connecting these cortical areas. Looking at the white matter connections wasn't possible in large number of people in vivo until recently, when diffusion imaging tractography was established as a research tool. In a seminal study using diffusion imaging, Scholz and colleagues localized changes in fractional anisotropy, a measure of white matter microstructure, in the parietal white matter. They imaged a group of people as they learned to juggle a complex visual motor skill. This study provided the first ever evidence for training-related changes in white matter structure in the healthy human adult brain. While this study looked at experience-dependent structural changes, we were interested in how a priori variability influences function. The first study looked at the variability in the arcuate fasciculus, a language pathway, and a correlation with the California Verbal Learning Test. In this study, they found 3 different groups in terms of the anatomy of the arcuate. The first group, as seen on the bottom left here, had the arcuate fasciculus only present in the left hemisphere, and it was not able to trace the arcuate in the right hemisphere. Group 2 was still strongly left lateralized, but had some connections in the right hemisphere, whereas group 3 was completely bilateral. So by just looking at the arcuate fasciculus, one wouldn't be able to say if it's the left or the right. Now, these groups were then correlated with their performance on the California Verbal Learning Test, and they found that the group with the more bilateral anatomy, which in this case is group 3, significantly outperformed the groups that were more lateralized to the left. We then brought this result to the clinic to see if the a priori anatomy of the arcuate fasciculus can predict language recovery after stroke. What we observed was that aphasic patients who suffered a stroke to the left hemisphere had a tendency to recover better after 6 months if they had the arcuate fasciculus in the right hemisphere. Recently, we expanded this approach to study primary progressive aphasia, a neurodegenerative disorder, and mapped the variability of the white matter and the cortical areas. In this study, we demonstrated that the variability in repetition deficits strongly correlated with a differential pattern of degeneration in the gray and white matter. These studies show that mapping white matter may be an ideal surrogate measure to capture inter-individual variability in structure and its functional correlates. In the last 15 years, diffusion tractography has become an established, non-invasive, quantitative method to study connectional anatomy in the living human brain. Tractography has widely been employed as a neuroimaging biomarker to link white matter phenotypes to cognition and clinical presentations. These differences are beginning to explain the observed variance in cognitive and behavioral measures. We have reviewed the use of tractography to study variability in connectional anatomy and its functional correlates. Looking at 326 studies, we observed that clinical cohorts, as in neurological and psychiatric patients, were most commonly studied, followed by healthy controls. By reviewing the literature, it is evident that variability in some tracts is highly sensitive to differences in behavior and/or clinical symptoms. The most sensitive tracts to correlate, as you can see in the figure here, were the corticospinal tract, the cingulum, the uncinate, and the arcuate fasciculus. When focusing on individual tracts, we can identify the cognitive functions that load on each tract. So what is the functional pattern of each white matter tract? On the left, you can see the results from our study that show that, contrary to common belief, there is no one tract, one function correspondence. For example, the corticospinal tract at the very top of the slide is most commonly correlated with motor functions or deficits, but also correlated with executive functions. The uncinate fasciculus, on the other hand, at the bottom of the slide, correlated equally with memory, language, and executive functions, and to a lesser extent with further functions. While our study had a limited range of functions, a recent meta-analysis of neurosynth data has included 590 functions and mapped them onto the white matter. This atlas is provided slice by slice and shows the complex architecture of functional white matter correlates. Now that we have seen that tractography is a sensitive tool to study variability in structure and its functional correlates, it'd be important to know how variable our connections are. And this is what we looked at in the next study. What you see here is the first ever map of white matter variability within the human brain. We compared the deformation of individual diffusion maps to a standard template to estimate the variability between people. If you want to know more about this map, you can find my other talk in the educational course on brain parcellation and functional territories, where I talk about this particular study and the methods in more detail. This map shows that regions supporting higher cognitive functions tend to be more variable, while the white matter in older parts of the brain is less variable. This is shown here, for example, by the arcuate fasciculus being more variable compared than the cingulum fibers. We attribute higher cognitive functions, especially language, to being uniquely human. So how variable is the monkey brain within its species and compared to us humans? One advantage of tractography is that it can also be acquired in non-human primates, which opens the door to learn from comparative anatomy studies. The methods for comparative anatomical studies are progressively evolving, and we have a comprehensive overview of neuroimaging methods in Michel's talk on imaging brain evolution, the next frontier. So I'm not going to go into too much detail here. All I want to highlight is that in recent years, tractography has evolved as a comparative anatomy tool, allowing us to study the in vivo anatomy on a large scale and across species. Tractography studies reveal some shared and some unique patterns of structural connectivity. I will present some recent tractography applications to study the variability across species, with a special focus on the parietal and the frontal white matter. Half of this image you have seen earlier when I was showing you the inter-individual variability within humans. Now we're looking at the variability of the whole brain white matter between species of humans and macaque monkeys. Similar to the map in humans, there are areas of low and high variability between the monkeys. There are also similarities between us. For example, the central white matter is less variable and the more superficial white matter is more variable. Whilst we have seen that the monkey brain was overall structurally as variable as the human brain has an additional left hemispheric asymmetry in variability across several phylogenetically recent regions. Most of these regions are associated with uniquely human functions, such as language, for example. This is demonstrated here on the left side by looking at the temporal cortical regions that are significantly left lateralized in humans, but not in monkeys. This implies that evolution may play a role in the development of the brain, and studying variability might be one way of tapping into this development. Now, this is what we can learn from a whole brain white matter analysis, but it's also of interest to study interspecies variability in subgroups of connections. I want to use the last few minutes and talk about two studies where we did focus on the parietal and frontal fibers only. Anatomically, the parietal lobe is at the crossroad between the frontal, occipital, and temporal lobes, thus providing a middle ground for multimodal sensory integration. Functionally, it supports higher cognitive functions that are characteristic of humans, such as mathematical cognition, semantic and pragmatic aspects, and abstract thinking. Across species, we compared similarities and differences in tract anatomy. For example, the trajectories of the white matter pathways and their cortical projections. What we found is that the largest tract identified in both human and monkey brains is a vertical pathway between the superior and inferior parietal lobules. The second prominent intraparietal tract connects the postcentral gyrus to both the supramarginal and the angular gyri in humans, but only to the supramarginal gyrus in the monkey brain. The third tract connects the postcentral gyrus to the anterior superior parietal lobule and is more prominent in monkeys. We were able to trace short E-shaped fibers within both species. In addition, a tract connecting the medial parietal cortex to the lateral inferior parietal cortex was only observed in the monkey brain. Overall, our findings suggest a consistent pattern of intralobar parietal connections between humans and monkeys exists, with some differences for those areas that are cytoarchitectonically distinct features in humans. In the next study, we looked at the frontal lobe white matter. That means all the connections within and beyond the frontal lobe across different species. And the species we looked at here is the human and the vervet and macaque monkeys. What we found is that the frontal lobe networks in the human brain occupied about 66% of the total brain white matter, but only 48% in monkeys. Among the long association tracts, the greatest difference was found for tracts involved in motor planning, auditory memory, top-down control of sensory information, and visual spatial attention, with no significant differences in frontal limbic tracts. In particular, tracts associated with language and higher cognitive functions are larger in humans, whereas limbic tracts are either the same or smaller. This supports the hypothesis that the emergence of higher cognitive functions in humans is associated with increased extended frontal connectivity. These studies have shown that tractography is a valuable tool to map variability in white matter connections between species and might give us an insight into brain evolution. And with that, I want to come back to my take-home message that we are all the same, but we are different. So what we have seen in this talk is that there is a significant degree of variability in the brain. We have seen that we can measure variability across species. Tractography has been proven to be a sensitive technique to capture this variability. And very importantly, this variability can be correlated with behavioral and clinical measures. We have also seen that variability may be linked to evolution, and therefore future studies should aim to measure variability consistently across people, across species, and using combined multimodal imaging. Thank you very much.
I'm Katherine Bryant. I'm a postdoc in the Cognitive Neuroecology Lab at the Center for Functional Imaging of the Brain at University of Oxford, and I'm going to talk to you today about neurophylogenetic approaches to human brain mapping. First, I'll talk briefly about human evolution and what that can tell us about specializations of the human brain. Next, I'll do a real quick overview of what phylogenetics is. And then at the end, I will help you combine phylogenetic analysis with neuroimaging techniques and see what we can do with those two methods together. So for those of you who don't know, these are some of our ancestors. And I put them in roughly linear order from their appearance from left to right. So this is going back maybe about 4 million years or so. And what you might notice is that bipedalism and other features that we associate with human uniqueness evolved relatively early. But as you go through this lineage of human ancestors, you'll notice that one thing that did change for this evolutionary period was the size of the brain. So that was one of the major things that changed, was the size of the brain increased. Another way to look at this is to look at the fossils that have been unearthed over this period. And from left to right, we're looking at 4 million years ago up to the present day. So on the far right, you see some of the very early human ancestors or relatives of human ancestors had essentially a chimp-sized brain. So they were about the size of chimps, and they may have been walking relatively upright, but their brains were not any larger than a chimpanzee brain. As you move in time, you'll notice that we see striking increase in the size of these brains. So during a certain periods of climactic variation that have been associated with this, we think that might have pushed some kinds of selective pressures to make brains of hominins larger. And indeed, if we look at the cranial capacity of these different fossils, which is one of the few methods we have to comparatively look at brain organization in humans and our relatives, you'll see that early brains, again, around 400 centimeters cubed, pretty small. And as Australopithecines changed over time, those got larger. So the Australopithecines had selective pressure to have larger brain. And then following that, the emergence of the Homo lineage, which is the genus that we're a part of, much even larger brains, until finally Homo erectus and Homo heidelbergensis started to get these, again, increases in the size of the brain that led to the evolution of Neanderthals and what became anatomically modern humans, which have brains around 1,400 centimeters cubed. So many, many times larger than our early human ancestors had. So one question this raises is, did the brains just simply increase in size, or did other modifications happen during this period? One tool that neuroarchaeologists can use is something called endocasts. And these are things which are basically fossilized matter that gets inside the brain case during the formation of the fossil, and it takes the place of the brain. And not only can you see things like lobes of the brain, which is already pretty impressive, but you can see from these images here, these are Australopithecine endocasts, you can actually see impressions of the different sulci and gyri. So it can tell us something about like how the surface of the brain has changed as the brain gets larger. With some modern digital methods and computational methods, we can take this further, compare these endocasts to modern brains and brains of extant species like chimpanzees or bonobos, and see what is the best way to model the differences in the brain. And here we see on the right, it looks like parietal modifications might be the major changes that we're seeing. So the brain didn't expand uniformly over the entire surface, but it looks like there was some selective pressure for parietal increases. And I'll come back to that later. But so these are the kinds of data that we can get from fossil information, but obviously we are limited because we don't have actual tissue. So that raises the question, well, if we want to know more about organizational specializations in the human brain, what can we do? And that points us to the direction of looking at other species that are alive now that we evolved in parallel with, and see what are the similarities and differences. So that brings us to phylogenetics, which is a method that you can use that's used in evolutionary biology in order to determine the relationships between different species. So many years ago, before we came up with the theory of natural selection, we viewed organisms as a sort of scale, at least in the Western world, in terms of simplicity at the bottom and complexity at the top, and that things were moving in a unidirectional way. Darwin and his contemporaries realized that this was not a good way to model the relationship of different organisms, that in fact it's a non-hierarchical, non-linear pattern of change over time that leads to branching and diversification and modification. Nowadays, with powerful genetic methods, we can build these really complex trees that can describe, all of life, for example, based on genetic markers. And we can build these great trees like this that can show us how things diverged over time and in what ways they're related to each other. So in phylogenetics, you basically zoom in on one of these branches and you look at it and you kind of can get a sense of how these organisms are related and use traits to try to determine the connection between them. So on the right-hand side, I have these ends of these branches labeled as species of interest. That's where your species would be. And then we can look at whether or not they share traits, and if they do share traits, how that's related to the most recent common ancestor. So in addition to identifying which species have similar traits, we can use the genetic information in the phylogenetic tree to determine what the most recent common ancestor might have had and when that trait might have emerged. So the way you do this is cladistic analysis. So we look for shared ancestral characters and shared derived characters or traits. So in a synapomorphy, we have shared derived characters. So these two nodes here in white represent two species that are sharing a trait that the other species in this family do not have. Therefore, the most parsimonious explanation for the origin of the trait is that it was their most recent common ancestor that evolved this trait. That's more likely than saying them both evolving it independently of one another, especially because they're right next to each other on the tree. The corollary of this is looking for ancestral traits. So if you find traits that many of the members of the family tree have, you can make a safe assumption that this evolved early in the lineage, and it's something that they all share because of that original evolution. Now, one thing that can cause some complications is something called homoplasy, and that's when two different individuals on different parts of a tree are showing the same trait. And this is something called parallel evolution or convergent evolution. So it can happen that a trait will evolve spontaneously in two different lineages. And in a tree like this, where you don't have much data, it's hard to resolve whether it's actually this bottom node is the common ancestor evolved it and the other one lost it, or they evolved it separately. But fortunately, we have very complex trees with a lot of data, and we can usually resolve that if you sample enough species. So let's go back to brains. So what's a way that we can combine neuroscience and these phylogenetic methods? So we already know the genetic relationship and the historical relationship of these different species. Now we can take their brains and try to figure out, well, what are the features that evolved when? So here, this is a very simple analysis where we're looking at the temporal lobe, which is arguably like a primate specialization, or at least primates have a pretty large temporal lobe. And so I was interested in trying to figure out what features of the temporal lobe might be unique to humans and which we share with other species. In this example, this is just, I'm marking the sulci on the surface of the temporal lobe, and you see that all primates have this blue line, which is the superior temporal sulcus, but only humans and chimpanzees have the red line, which is the inferior temporal sulcus. So that tells us that the most likely explanation is that the ITS, the inferior temporal sulcus, is evolved in the ancestor of humans and chimpanzees. Another thing you could look at all kinds of traits. You could look at association cortex. So here, this is a visual representation of the myelin content of the cortex using neuroimaging structural information. And we can use the thinness of myelin, or the lower myelin content, as a proxy for demarcating where association cortex is. So if we look at this, for example, we can see that both humans and chimpanzees have this an apparent expansion of this like blue, lightly myelinated association cortex, especially in the inferior temporal lobe and this inferior parietal lobule. Because they share that, and the macaques, which are my outgroup, do not have that, or at least not to that degree, we can safely say that this expansion of association cortex in parietal and temporal areas is probably a great ape specialization. Like our common ancestor with chimpanzees evolved it first, and then we inherited it from them. By contrast, there's a lot more what appears to be association cortex in prefrontal cortex in humans that I'm not seeing in these maps here with chimpanzees or macaques, and that tells me that that's probably a human unique specialization, something that happened only in our lineage. So in recent years, I've been applying this method to look at white matter organization within the brain, so long distance connectivity patterns and how those have changed. And part of this was sparked by interesting work that came out in 2008, 2009, looking at how unique the arcuate fasciculus was in humans. And we know the arcuate is very important for language, and it made sense that that may have evolved its special connections in the human lineage only. So I was wondering, well, what about other white matter tracks? Are those also modified, or are they all conserved? Maybe only the arcuate is the only one that's different and the rest are the same, or maybe there's other modifications that we didn't consider. So I was interested in that, and also I had been looking at the data, and it's clear that most data is humans or rhesus macaques, but I wanted to know what was unique about humans. And so I was looking at chimpanzees directly. Now, if I just compared chimps and humans, it would be difficult to tell what was actually impossible to tell what differences were human specializations and what differences were chimpanzee specializations. So adding macaques to this data set allows me to at least get an extra data point to help me resolve the phylogeny of which what's an ancestral character and what's a derived character. Now I will make a note, more data is better. So going forward, I'm going to be doing other primates, and hopefully that will give more data, because one outgroup is good, but more outgroups would be even better. So the method I'm using in the laboratory that I'm working at now, which is a method that the laboratory came up with, was creating these connectivity blueprints. So I'll show you that first. But basically, I created a white matter atlas where I reconstructed all the major white matter tracks in chimpanzees, which hadn't been done before. Did that directly also in macaques and humans, and then created a connectivity blueprint, which is a way to look at the surface projections, or at least the trends of connectivity between the tracks and the surface. So I'll just show you first what the anatomy looks like. So here's the arcuate, for example. And again, we see the chimpanzee arcuate is not nearly as extensive in its temporal connectivity as the human. And here's another example, just SLFs. And I didn't have a reason, for example, to think the SLFs might be different, but at least this early data that we have is suggesting that Humans seem to have a rotation of SLF2 in the anterior projections that we're not really seeing in the macaques or the chimpanzees. So we're already picking up some interesting differences just on the level of visual inspection of the way the tracks look. We also found interesting differences when we looked at how these tracks connect to the surface. So one striking difference you might notice here, so the top row is the macaques don't tend to have connectivity reaching the anterior temporal lobe, whereas the chimpanzees and the humans do. So to me, this suggests that in addition to the expansion of the anterior temporal lobes, that association cortex I showed you before, it also appears that there's been change in connectivity to that part of the cortex. So from there, from the surface data and these bodies of the white matter track data, we can now do a more complex analysis where we feed in all the tracks. There's 24 tracks from each species, and we compute differences between species. So the way we do that is we create a blueprint, which I showed you, which was the different tracks, and then we have one for each tract. And then we're able to compute a fingerprint for each single vertex on a brain. And it tells us what's the connectivity profile with every single tract I have. And then we tell it to search the space of another species and find the closest match possible for that connectivity fingerprint. And then to the degree to which it doesn't match, it computes a divergence value. So I'm going to show you some data that's going to show you a map of what parts of the brain have the poorest match with the other species. And that can give us insight into, oh, this area might be uniquely specialized. So here first, I'll show you the more, we have 30 million years of evolution separating us humans and macaques. So we expected to see some important differences, and indeed we do. So the red is going to be areas that have the least, the poorest match with any part of the macaque brain. And so in the humans, we're seeing especially hot areas posterior temporal lobe and up into sort of this the upper limit of like what we would call classical Wernicke's area. That's not surprising at all. It's kind of encouraging to see that we would have expected this area to be modified in human evolution. And we know the arcuate, for example, has new connectivity there. We also saw, interestingly, some hotspots in the superior parietal lobule, which we didn't necessarily expect from the neuroimaging literature, but based on the paleoneurology literature that was showing expansion of the parietal lobes that I showed you at the beginning from the fossil data, that actually wasn't surprising. I thought, well, maybe there's some important parietal modifications that we don't always think about when we think about human specializations. Finally, we saw some prefrontal differences, which again, I think we would have all expected, especially in Broca's area, but also up into the dorsolateral prefrontal cortex, which again, we would have expected. Now going to humans versus chimpanzees, this is the direct comparison there. We see a slightly different picture. So again, we see these massive differences with Wernicke's area, which is nice to see, and I think consistent again with what we know about how the arcuate is connecting up to that area. But we're also seeing, again, the superior parietal change, which again is consistent with this notion that perhaps Australopithecines, who were more chimp-like, may have had lower parietal bones than we do, and ours expanded. And perhaps when that expansion occurred, we also saw some rewiring of those areas in terms of connectivity. Interestingly, I saw less divergence in the prefrontal cortex than I perhaps expected, especially Broca's area. I mean, there is some, but not as much as I would have thought. So that, to me, suggests that a lot of the rewiring seems to be happening in posterior areas and in temporal areas. Another possibility is, for example, there's been some work, and I think Erin will talk about this a little bit, about parietal frontal networks being especially important in humans. And it could be that in terms of the parietal frontal network, some of the important changes that are happening are actually in the parietal area. So you can have changes here that will have impacts on how your prefrontal cortex works, but it may not, but perhaps some of the connectivity differences are not as dramatic in the prefrontal cortex in humans. We don't have to even stop there though, because we can also dig into this data, click on a vertex and look at what the actual connectivity profile is. And I'll just go very quickly over this, but basically we can find simple differences like here. It's mainly a change in SLF2 and 3 that's responsible for differences in the superior parietal changes in humans versus chimps. So that's just basically one tract that's making the difference there. We also can click on areas like this Wernicke's area that are showing very complex fingerprints that have complex differences with chimpanzees, which is nice. We replicate the arcuate data that we already knew was there. It was nice to see it again. But we also found that there might be some important stories to be told about what ILF and IFOF have changed, how they may have changed in the human lineage that we hadn't really considered before. given that the ILF is important for visual processing, that the inferior temporal lobe is really important for object recognition, to me this suggests that some important differences between humans and chimpanzees, in addition to language, may also be related to the what pathway, or the visual object recognition pathway. So what do I do with these data? So now I have these interesting things, but I want to synthesize them into my findings. So I can create my little tree. I can plug in the data that I see based on the shared and unique traits of each species. And overall, what I'm seeing as the major theme at this point in my research is that in terms of white matter connectivity of long distance tracks, the reorganization of prefrontal areas, I think, happened in human evolution prior to our divergence with chimpanzees. And then what made the human evolutionary trajectory especially unique in terms of white matter connectivity may actually be more centered on these posterior temporal areas and some of these parietal areas. I wanted to briefly just highlight some other work in our lab that's also using the same method. So here, this is work from Leah Rumuzeye, where she's looking at just in very close detail white matter tracks within the temporal lobe and using phylogenetic methods and finding some modifications to ILF that appear to be unique to the great ape lineage. So some of that ILF differences might actually be happening even prior to our divergence with gorillas, which was even earlier. So it may be a great ape specialization. And additionally, this work with Nicole Eichert, where she's working on how to basically manipulate surfaces from different species, which is a very challenging thing, in order to directly ascertain differences in how tracks are connecting to the surface, where we would expect them to be if it was just a simple expansion, and then calculating what those differences are and trying to then determine what's unique about how human brains have been wired. So finally, future directions. I'm obviously been focusing on primates, and I continue to do. I have more primates to look at. I have more outgroups that I want to see. I also want to look at smaller primates with like smoother brains to see, can I find some of those basic tracks in the same organization? What are the differences that I can find? But I'm also interested, obviously, in looking at other large brain species. So the interesting thing about species that have very high civilization quotients is they're species that have had the same selection pressure as humans in terms of a selection pressure for a larger brain. But they're in very different lineages. And as you can see from this dolphin and this elephant, the brains, at least on a gross level, look quite different than from a primate brain. So I'm interested to see, are there commonalities we can find in the white matter organization? What are the similarities? What are the differences? Are there different ways to achieve the same goal? What things are just properties of large brains versus things that are lineage specific? I'm also interested in expanding that to other mammalian orders, so you can make all kinds of hypotheses that you might want to test about how brains evolve. For example, you might be interested in carnivores because some of them have relatively large brains and they engage in complex social behavior. So maybe you'd be interested in doing that, for example, or how Erin is working on looking at domestication versus wild type, how that affects brain organization. You might also be interested in a species that has very different behavior, like an ungulate, artiodactylid, so herd species that are highly social, but we don't think of them as having sort of like complex goal-directed behavior, but maybe they have some similarities or differences that we wouldn't expect. So just to summarize, human brain evolution is characterized by brain expansion and as yet poorly characterized reorganization. If we combine methods from evolutionary biology with neuroscience, we can possibly identify what structures are unique to humans. And the neurophylogenetic study of non-primates with large brains, complex cognitive abilities, and other interesting behaviors can permit us to better understand the structural specializations associated with these evolutionary changes. And I want to thank, massive thank you to the Cognitive Neuroecology Lab. Rohir Mars is our PI, and other members of the lab, including Nicole Eichert and Leah Rumuzeye, and one of my collaborators, Longshuan Lee, who's at the Marcus Autism Center in Atlanta, Georgia, and all of my various funding sources. So thank you.